It's all yours, SFT.
Awesome. Thanks so much, guys. Tonight's the night. I want to thank you all in the chat for waiting patiently. This is going to be a really good discussion. I want to thank Ramat and Erica. Her YouTube channel is Gutsick Gibbon, so be sure to check that out. I want to thank them both. Enough for me, though. We're going to get right into it here. I'm going to pass it off to we can start off with Erica if you want to just kind of give a brief introduction. You can tell the audience, what's going on your channel. Maybe briefly go over your position, and then we'll hand it over to Ramat, followed by a discussion.
Sure, absolutely. All right, I'm Erica. I go by Gutsick Gibbon on YouTube. I cover videos on anthropology, paleontology, evolution, zoology, things like that. Mostly because I like it, but also because I have an education background in it. And today I believe we're just covering mutations in epigenetics. I imagine we'll cover the Italian wall lizards quite a bit in relation to which sort of model is more parsimonious with the evidence that we see, be it creation or the evolutionary theory. And I, of course, am in favor of evolutionary theory. And that's pretty much my stance.
Okay, well, I guess that's me then. Yeah, I mostly spend my entire life editing videos for Standing for Truth, so that's pretty much all I do right now.
That's why you get paid the big bucks, Matt.
That's right.
One day, hopefully.
With pretty much all the content we're putting out now, that pretty much keeps me more busy than you guys could ever imagine. I found it really fun, but now it's just, wow, it is overwhelming to make content for videos. So this is usually why I don't like to debate. I find making videos more entertaining and fun, which is good because Standing for Truth likes to debate. And sometimes I get dragged into it myself, like right now. So this will be a good one. The last one I did was on my specialty, which is gerontology and the study of aging. So this one is pretty cool because it's going to pit beneficial mutations with evolution. So this is going to be really good. And yeah, I'm ready to start whenever. Let's hit it.
Awesome. Okay. Well, thanks for the introductions, guys. Yeah, this is going to be a lot of fun. We'll keep it respectful. I think it's going to be a profitable discussion. So yeah, as Erica mentioned in her introduction, we can start off with the Italian wall lizards if you'd like to, since this whole discussion kind of sparked from that specific topic. And that might be just a good way to move forward. So whoever wants to start, go ahead.
Sure. Go ahead, Eric.
No, I mean, absolutely. I think that with the Italian wall lizards, so for those of you who don't know, and I imagine that those following this channel probably have at least a cursory idea of the Italian wall lizard sort of experiment and observations that go kind of in tandem with that, as well as the results from 2008 and so on and so forth. But essentially what we have is a population of Italian wall lizards, which were split between two islands. This population was initially omnivorous, primarily, usually, like the diet was mostly insects, but omnivory would be probably the best way to put it, as I believe there was plant matter involved, however small. But the population that was split into a new environment after several decades ended up evolving cecal valves, as well as a different head shape and larger sizes, in addition to behavioral kind of novelties and a penchant for eating plants, heavy herbivory. And this has been deemed, at least in the most recent paper that I saw, as sort of this incredible example of phenotypic plasticity. And phenotypic plasticity is very vital for the concept of genetic accommodation and genetic assimilation, which in and of themselves lend themselves to morphologic change. And so I would say that the Italian wall lizards, and I believe many would agree with me, evolutionary biologists and biologists of varying fields, that this is an excellent example of evolution happening rather quickly. Now, it should be noted that these lizards, once you, if you kind of take them out of the evolved lizards, that is, once you take them out of their natural environment and feed them a heavy arthropod diet, they'll actually lose the cecal valves, which is, again, very interesting as it is an example of phenotypic plasticity. But this doesn't change the fact that this morphologic range is a result of genetic change in this population through time. And that's pretty much what I have to say about it, if Ramat just kind of wants to take it away from there.
Yeah, absolutely. What happened in that 2008 study is they only gathered a few selection of these mainland lizards. So they saw the mainland ones and said, oh, there's probably no cecal valve in these, because every one that they looked at they were eating insects. However, in a later study in 2015, when they actually went and gathered all kinds of different lizards, they actually found that 19% of the mainland lizards actually have the cecal valve already. And the most fascinating thing is, guess what? They were eating plants as well. So anytime something is going to a plant-based diet, it seems that the cecal valve is required. It's just like a muscle that atrophies in a human being, and it goes in this one. The cecal valve is actually a muscle. That's the thing. It's a new muscular valve that they found. It's not just some new anatomical feature that popped up out of nowhere. Matter of fact, if you type in vegetarian lizards and how many exist, how many species, they find out 1 to 2% exist. Well, how many lizards actually have the cecal valve? Well, 1 to 2%. It's exactly in line with what we would expect when something actually eats the plant. We believe in our model that animals were created to eat plants in the beginning. Well, this would make sense, so they were getting the anatomical ability to eat plants, and this is built into their DNA already. So it can vanish when it's not needed, and it can come back when it is needed. So that was a pretty fascinating thing, but they can actually track all the way back to 1971, finding the cecal valve and things. That was found in another study in 2008, where I found that one. And they, like, within 15 weeks of a diet change, they completely dissipate this muscle. It atrophies up that quickly, but yet their offspring will still have it. It's just based on diet change. So it's already pre-built in existing thing. And what I find ironic is that you guys need hundreds of examples of these kinds of things, and the cecal valve is just one. I mean, just one. So that's not looking too good. I mean, we would want hundreds of thousands of examples of these new anatomical features jumping up, and they're just not.
So the thing, though, with that, Ramat, is that evolutionary theory or evolution in general of species is driven by environmental change and mutation. So if you have mutation, but you don't have a selection that's going to select for a novel change isn't going to happen, or at least that's not what we see. That's not what we would expect to happen. In fact, if we were seeing rapid evolutionary change in a stable environment, with no pressures to kind of drive that would run very contra to evolutionary theory as a whole. And I think I covered kind of some of the things that you said just now in your statement, sort of in my opening statement. I agree. It's phenotypic plasticity. I covered, that the fact that these cecal valves revert back to sort of a smoother gut once you feed them an arthropod diet once again. But the interesting thing is it isn't just the cecal valves themselves that lend themselves to this sort of evolutionary change. There's also a symbiosis or a mutualism going on, because when you look into the guts of these herbivorous Italian wall lizards, what you find is nematodes that are essentially helping with gut fermentation. So, and at least to my knowledge, and you can correct me on this if you have read the 2015 paper. I don't believe I've read it. I read the 2014 one, but I don't believe the mainland lizards have any of the nematodes present in their guts.
I haven't seen the 2014. No, I'd have to look at that one.
Okay, well, it looks like I guess we're at an impasse on that one, because I haven't read your paper and you haven't read mine. But I would actually kind of like to push back a bit further on that. I mean, I understand that you are kind of in the same lane as I am, with 2% of our sort of squamates having an herbivorous diet, and cecal valves as well. But you have to take into account too, like dentition and things like that. Like, I understand the link that you're making. You're saying, okay, well, these, lizards, these animals that we typically think of as carnivores or, insectivores at the very least, but some of them eat plants. I mean, you're not going to find any animals out in the wild whose diet contradicts their teeth. You know what I mean?
Actually, you can look at some copperheads and venomous snakes that actually eat kelp. You can they eat dulse. They walk around the islands, and they go around and they eat seaweed at night. And these are venomous copperhead snakes. They found that was interesting. They find they do it at night, so that's why they didn't actually discover this until recently. And
now, the question would be with that is, what percentage of their diet is the kelp making up?
Pretty much entirely. They believe that they're entirely misconstruing eating fish or eggs with the seaweed, because they smell like the fish. So the snakes walk around, and it's in our book. You can find it right there under all the different animals that decide to eat plants, like the shark that eats vegetables or the sharks that eat kelp, things like that. But the snakes were specifically eating it. It goes back to quite a while ago, the first snakes that were vegetarian that they found. And now their teeth are designed, as you would imagine, right? Their teeth are made for injecting and killing. But these snakes in particular are eating nothing but plants.
So I would be interested in seeing two things. One, I would love to see the link for that. I tried Googling it. Are you certain they're copperheads? I was, because I couldn't find anything with regard to copperhead snakes eating kelp. Are you sure it's not like a crate or a variety, some kind of elapid snake? Just when you get the chance, if you want to shoot me the link sort of in chat, I would love to take a look at it. But the reason I bring up teeth sort of in relation to gut is because it's those two things in tandem that tend to elucidate what an organism eats. And when I say teeth, of course, I'm meaning, the shape and the pitting and sort of the microwear on that tooth. And with the guts, we're looking at the actual formation of the gut, what the insides of it look like, and sort of the surface area of it as well. Because organisms that have high surface area in the guts, it's usually used to slow down plant material and things like that for fermentation. You usually see it in ruminants, not so much in monogastrics. But so I would be interested as well in looking at these particular snakes and looking at their guts and seeing if their stomachs actually match this diet that you're kind of referencing. As far as organisms eating vegetables, if you feed them to them, I'm not certain about which shark incident you're referring to here. But for instance, a great many animals will be kind of opportunistically omnivorous when given the chance. It's kind of horrifying that you can actually see videos on YouTube and things like that of horses opportunistically plucking up baby chickens and eating them. It's just kind of a very cruel side of nature, but it does happen. So I think that really what we're kind of seeing is you can kind of generally classify organisms as carnivores and omnivores, but again, as is typically true with nature and biology and indeed evolution as well, we're looking at general trends. So, if you want to shoot me that snake link, I would love to see it, or the one with the sharks or whatever.
Sure. But real quick, remember in our model, animals were created herbivore, right? So there was a natural instinct to be herbivore, and that's exactly what we see, because carnivores have to be taught how to kill. They have to be taught how to hunt from their parent. It's not instinctually ingrained inside of them. And teeth, they changed from epigenetics themselves, kind of like anytime that you change the diet. Matter of fact, when they took the Pottinger cat study and they wanted to change their diet, they actually didn't change the food. They actually started cooking it. That's all they did different. They started cooking the meat. And these cats, within generations, their teeth started changing and they had infertile offspring. It messed them up entirely. And to this very day, the zoos, it's illegal for them to cook foods and give them to animals to this very day. You can't do it at all. It messed them up genetically and it messed their mouth and teeth up. But
so a great many predators, though, have almost zero parental care, right? And they still do indeed eat meat, right? There are many carnivores, obligate carnivores actually, sharks and various elapid snakes, that and lizards, in fact, that have no parental care. No one teaches them how to do this. They do it based off of instinct or perhaps observation as well. But I don't know that I buy that. I don't think that that's correct, that they have to learn it from their parents, hunting.
Well, I would say it's best explained through epigenetics again, because this is what is inherited. If they electrically shock something and then the next generation, I'm sorry, I should explain it a little better. If they electrically shock a mouse when it smells fruit, they normally love fruit. But instead, the next generation of offspring is scared of smelling of fruit because of this electrical shock. So when something does something, it's inherited by the parent or by the offspring. So if the parent is hunting and killing, and that's what they do, therefore their offspring should do the same thing. So these are epigenetic tags. And this is what makes it so fascinating, because evolution needs beneficial mutations continuously, all the time. But every time a beneficial mutation comes up as what they thought was one, like the lactose mutation or the increased bone density or the HIV-1 or the high altitudes of the Sherpa people, you can go down the list. Every single one has turned out to not be a beneficial mutation whatsoever. And that's what's required for evolution, that every single one has been an epigenetic tag. So
two things with that. One, it should be noted that when we're referring to, I actually want to kind of pick your brain on this and see, what it is that you're referring to, but evolution does require beneficial mutations, but you have to understand what what we're talking about when we say beneficial. You know, what's beneficial in one context may not be beneficial in another, which is why environment is typically your primary selector. You know, it's sort of the molder of evolution itself. You know, you require mutation, that's the raw material, and then natural selection sort of shapes it. And that tends to come from the environment around. So I don't know why you would, you know, there's not necessarily an inherently beneficial mutation, at least not one that I would know of, that couldn't essentially be turned against the organism in another context. So things are very context specific. This is why, you know, I've had conversations with creationists a great many times who say, oh, you know, antibacterial resistance doesn't count as a beneficial mutation because that adapted bacteria is actually less fit than the other bacteria in the original environment. And my answer would be, well, of course it is. You know, it's only beneficial in this new environment, this one with the antibiotics in it. So I would actually like to pick your brain. What do you define as a beneficial mutation?
What would I define as one?
Yes, in your own words, like what do you,
Sure.
Matt, think is a beneficial mutation?
Sure. Well, the first thing I would have to say would be like, look at fitness. Fitness can't drive something into a new species. And that's all that we actually see in these experiments. We're looking for beneficial mutations that would add anatomical features and changes, linearly driving up evolution. So, for example, we have finally the very first experiment ever done on eukaryotic organisms, not bacteria, not Linsky's bacteria, nothing like that. And it's been done. I just linked it to you on the side. It's a eukaryotic yeast, and they grew 12 replicate cultures of this pure yeast strain that they gathered for 1,120 generations. Now, they didn't coddle these yeast at all. They actually went full bore on trying to mess these things up. So what they did is in the first dish for about 224 generations, they put them in a carcinogistic dye called Congo red. Then they moved it to copper ions, then an alkaline environment of like pH 8, then hydrogen peroxide, and then antibiotics. And then at the end of all these generations, the scientists concluded that beneficial mutations are undercounted in exchange for environment, both in the lab and in nature. They found that it was nothing but a loss of function and degradation of function. And these are what we are. We're supposed to be eukaryotic. So what I'm saying is that we need something driving the force of when we look at these charts and we're showing microscopic organisms becoming the tree of life, and we can't get even the single most primitive yeast organisms, which are eukaryotic, to change, or bacteria to change, or do the very first incremental stages, then that's horrible. I mean, you would need, I mean, when people say what is information, that's kind of like an umbrella term. You know, umbrella, it's referring to, you know, the entire genome, for example. I can give more of an example. I like to be more specific, but as far as what is information lost, think about it like the entire genome. One of you listening to me right now is missing over 100 million base pairs of DNA in their genome in different places. You know, they've been deleted through time. That's what we're talking about. That's why we recommend people look into the genetic entropy model. It's fascinating because we see loss everywhere. We see degradation. We don't see these beneficial mutations driving bacteria up into new things. And that's required. And epigenetics even makes it harder because no gene is ever changed. And the fascinating thing about the genes never changing, it's because when they compared humans and chimpanzees and they found that it's in the germline, meaning that it's permanent, inheritable between differences between human apes, meaning they can never be changed. So that's the fascinating thing is epigenetically primates have a different brain structure. That epigenetics alteration is different. Their methylation is entirely different in their brain and in their body than it is in human beings. Now, if we were the same as them and we came from a common ancestor, then there would be evidence of that because epigenetics would regulate the exact same way that it would in them because it's passed on in the germline. See? So
I would absolutely love to have a conversation about human evolution at another time. But do you think it would be fair to say, sort of just to summarize your statement, do you think it would be fair to say that a beneficial mutation is beneficial because it increases the fitness of the organism?
I don't know. If it seems kind of convoluted because that's the equivocation. That's like saying, well, evolution occurs because fitness increases. Well, yes and no. It increases at a loss. I mean, they're throwing genes out. Take the seahorse, for example, right? When the flood happened, is what I believe anyway, the part of the ocean got caught and put up into Mount Titicaca, 12,500 feet. Well, guess what? All the salt water almost dissipated out entirely at this point. There's still some, but for the most part, they only survived because they threw all their genes out that were important, that would have allowed them to actually maintain their life in the ocean. Yeah, their fitness went up, but at massive loss. They're completely blind. They're albino. They've messed themselves up. But yes, they survived and their fitness went up. But you couldn't put them back in the ocean; they would die. So the same thing with the Linsky bacteria. These bacteria that they pull out of these other places. That's what we see. Yeah, fitness goes up, but it allows the offspring to survive in that new environment. But they can never be moved back to their original. They get limited.
Right. So you know that is, though, just evolution, right? Like that's what evolutionary theory has always predicted, and that is indeed what we see. I mean, to say that you know, it's almost like I feel like creationists have this idea of like an ultimate fitness, which makes sense because in your model you would have sort of these Barrowman original kinds that are sort of perfect and herbivorous and free of disease and things like that. But the evolutionary model doesn't have that. There is no ultimate fitness for all environments that we sort of have as like the pinnacle of all. An organism can be most fit for its environment, but because the environment is always subject to change, those seahorses would indeed be very fit for their new environment, the new environment that they're in. And because they're surviving and thriving in it, that would mean that those mutations would be considered beneficial. Now, I think that that's a common roadblock that we come across when we're sort of discussing these two worldviews, is because the definitions in conventional science are very different from the ones that I see creationists use. I pulled up this paper and I was just kind of reading it and attempting to pay attention at the same time, which, you know, I'm not very great at multitasking. But I'm not sure. Were you attempting to say that the findings were that? Because I heard you use the word underestimate. Did you mean overestimate? Because this last sentence in the paper says these findings and the accompanying population genetic simulations suggest that molecular adaptation is consistently underestimated in nature due to its antagonistic fitness effects of mutations in changing environments. So, is it possible that you misspoke?
I'm unsure. Perhaps. What I was talking about is that the 224 generations per condition that were used by these people purposely didn't allow enough time for beneficial mutations to rise to 100% to become fixated. They wanted the cells in the flask to become basically the investigators. They wanted to see if the mutations were lost when these conditions were changed, and so they switched them time and time again, and in each for different length, and they found that many broken genes would pop up in the populations. So that's what
so you're suggesting sort of like a phenotypic plasticity? Is that why you brought it up with the Italian wall lizards? I'm sorry, I'm just trying to make sure I'm understanding you correctly.
Yeah, basically what I'd say is that the fitness is the total functionality of something. So when functionality goes down, we would say that it's a loss of fitness. It's kind of like a play on words. It's harder to define fitness because it's in our model, we go, well, look, it's losing information. It's losing genetics. Here, take extremophiles. Extremophiles live in the most extreme environments. That's why they survive there. But this is a small niche. And yes, they can live in the most abnormal environments, but they can never even move back to a normal environment. They're stuck there. And they're stuck in the niche. They've dumped out so many of their genes to allow them to survive. They're maxed out in what they can do. Kind of like Linsky's bacteria. They can never be jumping around. So if evolution were true, sure, you can change the environment again and they'll adapt even more, right? And therefore their offspring will come and therefore fitness will go up because their offspring can now live in that environment. But if that keep happening and their genome keeps shrinking and they keep throwing away genes, this is going the other direction. It's eventually they're going to run out of material like extremophiles and they're dead-ended. They're at the dead end.
Okay. I think I got where you're going with this. So you're essentially saying that because you're essentially limiting organisms to mutations that are deletion events. And you're saying that organisms can adapt to their environment, but this is going to be a deletion event, and eventually they're going to run out of genetic material in order to adapt to their environment, and then they're hosed. Is that kind of what you're saying?
Yeah.
Okay. So, but that doesn't happen because we have duplication events and duplication mutations and events, and as well as frame shift mutations and events and things like that add genetic material. And I know that creationists don't typically like to think of that as new information, even though conventional science would deem that new information. Sort of in the same way that it's like I use the example of the sad cat sat, right? So if you had a word that says the cat, and then you have a duplication event, so it says the cat cat. And then you have another duplication, so it says the cat, cat, cat. then you have a series of other mutations that change little letters here and there. You might end up with the sad cat sat, and that conveys quite a bit more information than simply the cat, right? Even though it's using essentially a very small number of letters and a very small amount of mutations. I believe it's like 4 or 5 actual changes, including those duplication events at the beginning.
Yeah, but they say, you can even research this. It says gene duplication is not a common event. According to evolution, it's 0.01 gene duplication per million years. And gene duplications are what? They're duplicating already pre-existing material. So if you're throwing out genes, eventually if there needs to be a duplication, it's not going to be there. It'll be kind of messed up.
But I think you're minimizing the sample size. I mean, you're basically like, I get where you're coming from, and it sounds quite intimidating when you say it that way. When you say, well, we're talking about, I forget exactly what you said. I think you said a mutation or a duplication event every million years.
Right.
But then there's also, with the vast majority of species, we're dealing with, I'm so sorry. Hold on just a second. I'm not sure where this music is coming from.
No problem. I sent you a link on that as well. You can see in the private one. It's the International Journal of Evolutionary Biology.
Hold on just a second. Can you guys hear that background music? I'm not sure where that's coming from in my house.
It's quiet though.
Okay, all right. If it's okay, I'll just talk through it because I'm in my dad's office and I'm sure he's just like turned something on in here. Yes, duplication events can be quite rare. But again, what I'm saying is it's not problematic because we're dealing with a vast sample size. I mean, if you have let's say we're dealing with the bacteria in an abiotic, or rather abiogenesis style event, right? So it's like you're at the dawn of time. We're talking like very, very like Archean period, sort of 3.8 billion years ago. And so you have a ton of different bacteria, and they're all attempting to undergo mutations simultaneously. I say attempting; they're all kind of going through their life cycle and things like that. It can be one mutation every million years, but it's one mutation every million years for billions upon billions of bacteria, which greatly increases your potential for sort of seeing this mutation. Even more so if it's selected for and it passes it on or divides even a single time, because that also increases the potential for this mutation to sort of proliferate out into the population. You see what I mean? So it's not like these events can indeed be rare, but I think that I see this quite frequently, like creationists kind of put it in a box almost and say, it's so unlikely that, that this could happen. Like I've heard one with sort of evolving new proteins or something along those lines. And it's like 10 to the 40th to the 20th or something like that. But that's evolving an entire lineage completely by chance, which is not what evolution suggests. Like pressure is one of the, natural selection and natural pressures is one of the most important things to evolutionary theory. It's like one of the tenets, you know what I mean? So I think that it's quite easy to make evolutionary theory sound very improbable, or even impossible, but only when you're using, half the pieces in the jigsaw puzzle.
But you did realize that you did say that requires deep time. That's it. Yeah, because
absolutely
it's required. So here, I'll give you an example. Take Neanderthal. We say they were, I do. I'm going to say they're pre-flood people. And what's fascinating about them is that they have more haplogroups. They have more blood types. They have more genes. They have more complete base pairs in their genome size. They have less total mutations overall. And that is exactly what our model is showing. We show that humans today are less than what we were in the past. Now, that's fascinating because that lines up with exactly what we would expect. Now, if a primate was doing the thing, all right, take out human genes. How many do we have? We have about 19,000 genes in us. Do we know that number is going up? No, if anything, it's going down. Every time they do an investigation, it's like, oh, we have less genes than we thought we did. And the number is plummeting each time. It's not because they're discovering that we're losing genes. No, I'm just saying that the number that we have isn't going up. And now that we know how many genes humans have, we're not going to find new genes being added. So this process of gene duplication and different things that is required for evolution, we just don't see it.
Okay, sorry, I'm trying to mute myself and unmute so you guys hear as little of this music as possible. I figured it out. It's my sister upstairs. She's playing music over a loudspeaker. Yes, so with regard to mutations and sort of, I'm imagining you're kind of suggesting that we should see more duplication events than we do, just for the sheer fact that there's so many humans and we've been around for at least a decent period of time. You mentioned Neanderthals, which again, I would love to have a longer conversation about human evolution, because that's kind of my wheelhouse. But the thing is that, again, like we're missing a piece of the jigsaw puzzle, right? I mean, duplication events work, right? They do occur, but they can almost pass under the radar if they're not selected for. They're not going to proliferate unless there's a reason for them to do so. Typically, gene duplication events, they do occur in human populations. It's just that, these enormous, vast changes that we find are almost, they're anomalous. They're not necessarily selected for one reason or another, because humans, as of right now in our modern state, are effectively free of all natural selection. We've kind of out-evolved that, fortunately or unfortunately. So that's why we have, immunocompromised people, etc. You get what I'm saying?
Yeah, yeah. I noticed that you like well, you would agree then that, or not agree, I was just saying, so you believe that a split occurred and humans evolved from a primate family. I've seen your videos and I've seen you debate on this, so you definitely agree with that. And have you seen the latest genetic study that showed that they've taken hundreds of thousands of species and they tested them and showed that they came out of a clear bottleneck that happened not too long ago, and that all species arose at the same time? Everything from anchovies and fish and primates and humans, all separate?
I have seen this study. This study was, I believe it was from about a year and a half ago, I want to say, because I used to have a coworker who was a younger creationist, and he brought this up to me. We had a lovely conversation about it, actually. But the thing is that that's again, it's a misconstruing of the actual situation. Of course, modern species are indeed quite new. The problem with that is that, again, speciation occurs almost constantly. I mean, we see this sort of gradient of change occurring in almost every genera. So when they say, like, I encourage you to go and look at that paper, because when they're speaking about what they're actually finding, right? What the findings actually say, what the data is suggesting, isn't that all organisms emerged very recently. What they're saying is we have a very recent range for the species that we have currently emerging from previous species. And the way that they know that is because you can actually trace that back genetically, right? I mean, I understand what you're saying, right? That it sort of seems like, but again, I would encourage you to look at the study, because it's not all species, like you said, from anchovies to artichokes or whatever. It is a great many of them. It's like 90%, 92, I believe, something like that. But interestingly enough, that also ties in with a rather recent sort of worldwide event, right? We have these interglacial periods and glaciation events. So I don't see that as problematic for evolutionary theory.
Well, you're actually in luck. I know that study like the back of my hand, and
Okay,
it actually is going to answer your question that you always ask people: where do we draw the line in phylogeny? Because
yes, I certainly hope you'll answer my question.
yes. Well, now, the fascinating thing about this is that study used DNA barcoding to do it, because it's down to the species level. Now, yes, I'm not saying that those species arose out of nowhere at the same time, because that's like Genesis, right? The creation of everything. No, this is the bottleneck of what we say is the flood. That's why it did actually count fish. So matter of fact, the person that actually looked at the study said, I can't believe it. I can understand maybe a human and some deer being destroyed in a worldwide bottleneck, but he says, but anchovies and herring too, it didn't make any sense, because even aquatic life died. So I did read the study very in depth, and check this out. What they do is they looked inside mitochondria, and they found that there's 37 genes. And the interesting thing about it is one of them is called the CO1 gene. Now, this gene is very important to all species, so much so that it's lineage specific, and it's called what's called selective sweep, meaning it passes on to every single lineage of a species in that family down the line. That's how important it is. So it's in you, it's in me, it's in primates, it's in fish, it's in everything. So the really cool thing about this is when we stand back and we look and we want to compare something, we don't just go, well, look, everything with hands is related, or, oh, everything with mtDNA is related. No, we got to be more specific, because when they tried doing that with just mtDNA, they found that domestic dogs differed by as much as 0.2% with wolves. But when they compared wolves, they found that there was 4% difference There means there's more differences between wolves than there is actually between wolves and dogs. So they needed something more specific. So they went directly into the DNA barcoding with the CO1 gene, and they found that it's always related to a family and it branches out into new species. Now, why they like it is because they can identify new species when they pop up, meaning the barcode shows up as a gap between something. So what I mean by that is that there's just a little bit of variation when something pops up as a new species. And so there should be low barcoding gap differences between something that just speciated and that's related. The bigger the gap, the less they're related, or they fall out entirely of the group if they go over like 60. So what happens is now we have a way to identify not only species, but what is a kind, because it works on a family level. So they went in and they studied Neanderthal, Denisovan, and modern humans, and they found low barcoding gap. So much so that the gap was only 1 to 4 barcoding gaps. Now that's nucleotide sequencing of this genome. But when they tested primates, they were 131 nucleotide sequence gaps between them. That's huge. But related to each other, they're only around 10. So I made you actually a video on this. It's actually going to be, I'm working on it, so it'll be out soon. But regardless, it's finally a way for us, besides orphan genes, taxonomically restrictive orphan genes. It's different than the other ways that we've been trying to identify kind. We now have like a trio. We have the heat maps, we have the DNA barcoding, we have in fertilization, things like this. So anyway, I talked too long. Go ahead.
So I want to ask you, what you just said was that humans, Denisovans, and Neanderthals were very similar. Yes, you would indeed expect that. And the reason is because we find individuals, as I'm sure you know, there is a difference between finding a fossil that is legitimately bone and finding, mineralized bone. Actual bone you tend to get DNA from quite a bit easier, which is why we've been able to sequence the entire Neanderthal genome. And that's how we were able to know that the remains of this individual, I think this study came out, I want to say it was like the past year. But we found a hybridization event between Denisovans and Neanderthals. So, naturally, yes, I don't think you'll find an anthropologist on the planet, even prior to that study, who would say that humans could not interbreed with Denisovans, Neanderthals, even as if you wanted to take it that far with Naledi or Floresiensis, although there might be some difficulties getting there geographically. But the thing is you would expect those differences to be indeed quite close, right? I mean, they're, for one, even in conventional science, they're still members of genus Homo, along with Ergaster, if you want to count Ergaster, Erectus, and Habilis. And again, what you mentioned this, I wrote this down because I wanted to ask, and I'm a little bit sick, so I'm a little foggy brained, but what primates did they test? That's a very, very, very important question. Because my question, to kind of reiterate, my point is that, it kind of falls down the line. This is kind of my version of my go-to question, as you know, because you've seen my debates. But we use genomic comparison between humans to tell paternity, right? And it works, and it holds up in court. And we use a very similar process, albeit a bit souped up, to make comparisons between species. And so my question is more on a technical level, right? And it's like, all right, well, where and or why do we draw the line, or do creationists at least draw that line between species? And why is it reasonable? And that's why I asked which primates, because you're going to get a very, very different result if you use a loris or a galago or a tarsier than you are if you use an orangutan or a gorilla or a member of genus Pan. So do you know off the top of your head? Or also, I would love it if you would send that study to me in the chat on the side so I can have it pulled up.
Oh, sure. Yeah, they tested the bonobo, the chimp, and the gorilla. That's I know off the top of my head.
Okay.
They liked it even more. Oh, so go ahead.
Yes, so I was going to say that would be perfect. That would be your perfect individual. So now we need some scale, right? We need to know if you tested the bark. I'm assuming I'm using the terminology correctly. I haven't seen the paper, so I don't know. But so now you want to establish a scale, right? You would want to say, all right, well, how do humans compare to mice? How do humans compare to cats? How do humans compare to insert mammal here? Because the general consensus, right, the trend that we see is no matter which area you compare, even if, let's say, you take a random protein and humans and chimps show a 25% similarity, that is indeed very different. But the sort of stipulation with human evolution is that that similarity is still going to be the closest between those two, humans and genus Pan, than it is between humans and any other mammal, similarly with chimps. So do you know, by chance, off the top of your head, since you know the study, if they've tested other mammals and what they got?
Oh yeah, matter of fact, yeah, they did. Matter of fact, they tested bear, caribou, all kinds of things. Matter of fact, that same study that I was telling you about earlier, where they tested the like 100,000 species, separate species, that was the one I'm talking about as well. This goes in conjunction with that. So that they've tested all kinds of things with this DNA barcoding. Tons. And you can see the visual layouts that they do for DNA sequencing, and they also test base pair combinations. So what they do is they look for sequences and insertions, and they try to combine them in multiple different ways. Then they found nucleotide sequences with the cheapest and fastest, because you can kind of stand back and it looks like a puzzle. You're like, oh, I can identify that's all fish. And then they just throw them over there. And then all these are birds. They throw them over there. And then they go, okay, well, now we got to get into the detail. And then that's where they have to sometimes go to base pairs. And but the CO1 gene is down the line used because it's the germ line. It's passed to everything in the species, and it's handed down. So if we were going to be taking that back to a split, an ape ancestor, it would have gone to both of our lineages. So we would share a lot of the CO1 sequences. But because the sequences are so vast and the barcoding is so big, they fall into an entirely different family. That's what's fascinating about the study on my end. So take for the COI sequences contain about 650 base pairs in most groups tested. As I said earlier, the human, Denisova, Neanderthal, all the way as far as you can go back in the human lineage, are only 1 to 4 nucleotide sequences different. And that shows that they're close related. So when we do the same thing with chimpanzees and bonobos, which are in the same family, again, there's more genetic diversity even between them at 58 nucleotide sequences difference. But anything that really goes over a limit, you see where they fall out of the family line. And so it's really good. You'll be able to visually see it in the video that I made for you. So it's nice to have visuals. Unfortunately, in a debate, you're just looking at my face. So.
Oh yeah, no, I know how that goes. So I want to again kind of pause it. Perhaps it's just my foggy brain with this, but so are you suggesting that because this? Let me just kind of frame it the way that I'm kind of picking up on it. So you're saying that based off of this DNA barcoding, you get genus Homo, right, and all the members of genus Homo sort of in a group on their own. And then you also separately, by quite a great many differences, you get the hominoids, so your other apes. Would I be correct in saying that?
Right.
Okay. That's what's expected, though. That's why we're in a different genera than the members of genus Pan and the members of genus Gorilla and the members of genus Pongo. So, and I would say, okay, kind of like, yes, I agree. I don't think that humans are literally chimps or that we should be in genus Pan. And in fact, again, every single member of our lineage we should be closer to than we should be to chimpanzees or bonobos, right? Because they're sort of like cousin. We're not related to them in a sort of linear fashion up and down, but rather we're at the end of a branch that split quite, some 7 to 13.3 million years back. So I would say, to kind of clarify, that a great way to figure out whether or not this is going to work for creationists with sort of this idea of kinds, right, is there a way to create sort of separation lines, right? Because the problem that creationists have, and I would say that Jensen has kind of attempted to solve this with this idea of like common design, amphibious vehicles for the transitional species, like because we create amphibious vehicles, God might, and that's why we see transitional forms, etc. Jensen likes to cover his bases. But I think that you're still kind of in trouble, right? Because, and again, please do send me this paper in the chat if, while I'm talking, if you get a chance, because I would love to have it pulled up. But you have to be able to have something to compare it to, something that, at least according to, like if you're attempting to disprove evolutionary theory, right? You want to have something that's going to falsify it properly. So what you would need to have is humans, muscelids, right? Cetaceans, felids, whatever, a bunch of different family members or group members, genera, whatever, and line them all up, and then compare it with this DNA barcoding, right? In order to see who's closest to who in sort of how you just mentioned it, but across a very wide range of living organisms. And then it would be quite interesting, and that's why I would like to see the paper, because I want to see what they concluded, to see if that matches what we find with regard to just comparing coding base pairs, through genomic sequencing, or sort of phylogenies that we get with molecular clocks and things like that. You get what I mean?
Yeah. No, they did test hundreds of thousands of species, so they've got what you want. There's two studies. They've got everything you want. They do line up fish, whales, but remember, we're talking about a species line. So when we're looking at the nucleotide sequence differences, 10 or more place them into a different taxonomic family. And when in terms of base pairs, any gap under 60 is related. So that's how we're looking at. We do have a different way. We do have a way. So when we're looking at base pair sites, you see humans alive today are never more than 2 positions different, exactly the same as Neanderthal and Denisovan. Yet chimpanzees differ by 60 sites, and gorillas are separated from 70 sites from us, placing them into an entirely different family. And there are 132 nucleotide sequences separated us. So not only are they base pair separated, but they're also, so if they were same in species like us, if they were still branched out, that missing link common ancestor would have passed on these because they're germline, like I was saying. So we would be able to see them in the line. And they tested all kinds of things. That original study where they tested 100,000 species, that is tested using DNA barcoding. So they did that as well. And the best thing of all was wolves. They tested wolves, and they looked at, like, are they related to dingoes and foxes? And they were related to all the different types of animals. But yet when they compared them to bears and badgers and raccoons and things that are supposed to be related to them as well, totally different outside the family.
Right, but you'll forgive me if I say this sounds exactly like what phylogenetics is. I mean, this, to me, like, you're saying this, and I'm hearing you, and I'm like, okay, so they're using genetics to group organisms into categories. Right, that's what we've done. That's why we have the caniforms, and that's why we have the primates, and that's why we have, sort of these groups that we have in zoology and when we try to classify organisms. So what I'm hearing here is that this is another means to do so. And that even by this means, we're still ending up with humans incredibly close to the other apes and closer to them than any of the other mammals, and that the apes are also much more closely related to the humans than they are to the other mammals. So I would say it sounds like a very fascinating study. Again, I'd really like to see it. So if you can, send it my way. But I don't understand the point that you're making. I mean, again, maybe it's me. Maybe I'm not understanding it. Or let me try it a different way. Are you trying to say that because we can group them into these categories based off of germline mutations, and I'm imagining that it's probably like a divergence or speciation time that they're sort of calculating, or rather looking at these lineages. You're saying that because we can group them into these different categories, those are the Barraman groups that creationists should use. Is that correct?
Yeah, sure. A Barraman is kind of like invented by a creationist, so I guess we'll use the term, sure.
Yeah, I mean, I'm just trying to use the right terminology. The terminology that I've seen in creationist literature tends to be created kinds are Barramans, right, or Barramans. Maybe I'm not pronouncing it correctly. But again, yes, I would agree with you. Organisms are going to fit into neat categories. Neat category. Neat is a very arbitrary term. They're neat enough to draw, nice trees to the best of our ability. But the problem comes with when you compare them to one another, right? Like, my problem is there's no stop point, because the very fact that they could do this comparison and that they could say, well, look how similar humans are to the other hominoids, and look how similar all of the hominoids are to one another when compared to all of the caniforms, which are all very similar to one another, but quite different from the primates. You know what I mean? So again, it sounds to me like this study is, it's giving a sort of a different way to categorize organisms, but it's just confirming the previous way. Sort of like isochron versus other methods of radiometric dating. Like, it's sort of a corroboration, if anything.
Well, the good part about this is that it's not just like a hierarchical pattern, because we predict the same thing, but this one is shows those clear genetic boundaries. And you'll see from the researchers what they say. They go, I can't believe this. It's astonishing to them because it's actually not what they predicted. It's outside of their prediction, which is really good, because this is also like I was saying, it's down at the species level. That's what they want. But that selective sweep mechanism that shows that this CO1 gene is passed down throughout the entire family line would actually show the relation of things. And what's really cool about this is it shows that the hierarchical pattern that's in humans is always in humans, and it should be the same that would be in the primate lineage. That's what I'm getting at. Just like you can follow it in the dog back to a fox to a wolf.
Right, but see, I guess I'm getting confused because I feel like you're mixing terms up a bit. So when you say that you're tracing this down through the family lineage, do you mean like the actual category of at the family level?
Right, exactly. You can go up to the family level with the barcoding, because
but not the order level, which would be primates or primates.
I would imagine that it's probably too small for that because it's 650 base pairs, but I'm going to find out as much as I possibly can studying this, so like I can get the video absolutely completely drawn out, and then I'll be able to differentiate of where I can go, because it's used for plants. When it first happened, it was like, oh wow, we can finally identify new plant species, because looking at a plant is extremely hard. Looking at an animal is a little bit easier. You can kind of differentiate the dog kind or the cat kind pretty easily just visually, but a plant, now you're getting really, really tough. So
but even visually, it's a completely bunk method to look at vertebrates and try to compare, or invertebrates, really any member of Animalia, because you've got very, very, very consistent sort of convergent evolution between forms if they live in the same environment. That's kind of what I was saying earlier. That's why you have in areas where there aren't sort of organisms that fill in the cat niche, for example, like in your traditional carnivore, right? Like on the island of Madagascar, you have fossa, right, which aren't strict. They're not the same as like a lion or a leopard or anything like that. They're entirely unique, but something had to fill the niche. Or the Tasmanian tiger over on Australia when they were still around. Marsupial, very not a canid, definitely not a canid, but looks very much like one. So I'm very much looking forward to your video. I want to see what you have to say, because to me, and again, it could be that I'm totally missing the mark here, and or maybe I'm taking crazy pills, but I feel like what you're saying is that this study is just a different way to confirm the sort of phylogenetic tree that we currently have.
Well, it's one way. It's just one. What I mean by that is you guys want the answer to the phylogeny challenge. So what we're doing is we're combining. We have the DNA barcoding now. We're going to have the taxonomically restrictive orphan genes. We have the statistical tool called heat mapping, which catalogs it compares the expressed proteins of species, because it proves by visualization methods that you can group species that belong to, physical comparisons by proteins inside of them. So we have that. Then we have zygote fertilization, which obviously is really good, but becomes ethical problems as well when you get into humans and primates. But we can use all these different varieties now to definitely be able to define what a kind is. We can do that with all these varieties rather than just a single heat mapping.
10 minutes left.
Yes, I absolutely look forward to hearing sort of the idea behind that and seeing what you guys come up with. But the phylogeny challenge is Aaron's. It's not really mine. I don't have a name for my challenge because I'm not big enough to have a challenge name. I just kind of it's just the question that I frequently pose.
I think where do you draw the line on phylogeny is basically the same as
it's kind of just limited, right, to primates. It's looking specifically between humans and chimps. And to me, because essentially what it sounds like you were saying, again, this is my best grasp at it, but it sounds like you were saying that via DNA barcoding, they looked at all of the primates and they got all of the primates in a single group, and they found that humans, Denisovans, and Neanderthals were very, very close together, and that the members of genus Pan, so chimps and bonobos, were really, really close together, and that they differed by 60 with humans, and then gorillas differed by 70. And I would guarantee you, if you looked at that paper, it would say that the pongids or the orangutans differed by quite a decent bit more, but not too much. Which again is exactly what we get with genomic comparison. We find that humans and chimpanzees, and as well as bonobos, are one another's closest living relative. I bet you too that if you looked at that study, I don't know if they did this or not, but that if they compared chimpanzees and bonobos to humans and then also compared them to gorillas, that they would have a 60 difference between humans, and probably likely a greater difference between gorillas, because that's what we get with the genomic comparison. And so while I appreciate the idea that this is going to, and I hope certainly that I'm very much look forward to analyzing to the best of my ability what you guys kind of put out as far as the kinds go. But this sounds to me like it's still putting humans with the rest of the hominids, or hominoids rather, particularly because you mentioned the caniforms. You're talking about all the canids, and you're saying, well, this is how we know that the wolves and the dingoes and the foxes and the dogs are all landing sort of in the same kind. But if you're going to use that methodology, and I would be willing to I'd put $20 on this, mostly because I'm poor. But I would bet that if you use that same methodology, Matt, that you're using to group those canids for kinds for the baraminology purposes, if you apply that to the primates, you're going to end up with humans with the rest of the hominoids.
Well, if that was true, then the dogs would have been lining up exactly with the other bears and the dingoes and the other things that were branched off from their lineage. Because remember, everything goes back to another family. So
they wouldn't line up with the bears. I mean, what you said was that the canids all lined up together.
Right.
So wolves and dogs, right, are, if I remember correctly, wolves and dogs share, if we're looking at just like a basic genomic comparison, they're about the difference between humans and Neanderthals. But I would wager that you would put all the cats together, the cat kind, right? And the difference between a domestic house cat and a lion is about 95.5%, I believe, when you're doing a genomic comparison. I can give you sources on these later, but obviously I don't have it right off the top of my head, which is much greater than the difference between humans and even orangutans. So, that would again put humans in, like, what I'm asking for is an empirical reason to separate humans from the other apes. And I don't think that this provides one. You know what I mean? Like, if you want to create kinds, I would love to see the different kinds. I'm saying that I don't think it's possible, as someone who's versed in anthropology and primatology. I don't think it's possible by any means to use the same methodology for every group of organisms to find your kinds and exclude humans from the apes. You might be able to say, we're using this methodology for all the animals that we don't deem special, in the image of God, and then we're going to use that categorization to separate the humans. But that's not empirical, so it's not science. You know what I mean? So I look forward to your video. I'm interested in seeing how you put humans separate from the other apes.
Sure, yeah. I'll make the video a little bit longer because I did leave out, orphan genes and different things like that, and where they're related. And humans are mostly in the brain, and apes are in a different spot. And I think that because humans have been thrown taxonomically in with primates, that it kind of befuddles everything, because they don't even look at the differences. They don't talk about the different ribs, the different how many spinal columns we have, the subcutaneous layer of fat. There's so many different vastness that would have happened in. You would think we would have kept some of the beneficial attributes that primates have physically, but we can get into that later. Another thing that you wanted to talk about one time was radioactive decay. And
yes,
that's fascinating, because we are kind of out of time, but I did want to talk to you about something very fascinating. They believe that stars explode and they create all of the different elements. But what we've actually found is that the prestigious Science magazine, they admit that supernovas cannot make any element over iron, which is, I think, like number 26 on the periodic table. Anyway, that's over 60 elements that cannot be made. But the Proton 21 laboratories in Ukraine, they created the events that replicate Noah's flood by Walt Brown's theory. And guess what happened? They made every single one in a process called Z-pinch. And it occurred quickly, not over millions of years. Instantly, they were able to make, matter of fact, they created more than the 60 elements. There was 3 that they don't even have a name for. And here's the interesting thing. The elements occurred, and when they were formed, exactly like they are on Earth today. And when they were tested old. So even if the clock, sorry, go ahead. You want to say something?
I want to let you finish. Go ahead. I have quite a bit to say on that. It'll probably have to be a brief conversation. I know we don't have that much time, but I want to let you finish your thought. I interrupted without thinking. Sorry.
That's okay. I just saw you starting to speak. I just don't want to.
No, I jump the gun sometimes. I'm sorry.
But okay, so a guy named Adam Blanco or something, he came up with a process called cold repacking. He found that super heavy elements decay into the other 94 elements that are relatively stable, but they also make the unstable one, the radioactive elements. We believe in our model that there was no radioactive elements in the pre-flood world, that they occurred during a flood. So what happens is, in this experiment, they wanted to find out like what could happen if this occurred. So they took lightning and they they coursed it down, and they did it. They actually did it by accident. So that's pretty cool. And what happened is that exactly the same proportions that these elements exist on Earth are exactly what happened. So that's strong elements that these formed during the flood, because their decay rates match exactly what we see when they form right in front of your eyes. Now, let's say you find a clock on the ground, and or 100 of them on the ground. You take that and you go, well, they're ticking at this pretty much constant rate. So you can guarantee, well, they're constant. We can guarantee that. So that's the cool part about this, because we can extrapolate that back. Like, well, when was it made? Well, you can't really do that. So, we can't tell when a clock was made just based on when it's rotating. Well, that's what we're doing with the elements. We believe that they started out at 0 or at 100%, and they've been decaying by half-lives ever since. But this method has actually extrapolated that that's not the case. They don't have, even if you start out with a 100% parent isotope to decay from, the proton laboratories showed that new elements are not like this. So that's what's fascinating about this experiment. They've showed our model completely.
Okay, so one, radiometric dating, there are many different methods. I recommend this book to everyone that I get the chance to, particularly those who are religious, it's called The Bible, Rocks, and Time. It's written by two, I'm not sure if they're old Earth creationists or theistic evolutionists, but they go through it's a dense book. There's like 100 pages just on radiometric dating. And I find it a very handy guide for things like this. You mentioned that I really want to see some of these studies, because unless they're quite recent, like as in the past 3 months, and maybe they are, to my knowledge, every time we've seen rocks form, right, on planet Earth, be it in sort of volcanistic conditions, which would, I imagine is most of them, they always start with a heavy bias towards the parent. And when I say heavy, I mean they start with all of the parent and none of the daughter. That's why, isotopic decay or radioactive decay, the law of radioactive decay, is sort of that spontaneous decomposition from parent to daughter. And it's not been violated in meaningful ways. Even the creationists had to admit this, right? So I'm interested to see this Ukraine study, but also I'd like to point out that the method of radiometric dating known as isochron dating assumes nothing about the parent material, and yet it still corroborates methods that do. Which would be to say very, very, very old rocks in, the by the methods that we're using for older rocks, and so on and so forth for younger ones, and gradient and etc.
Right, but that element was actually created over iron, so that would have been something that also formed during the flood. And by the way, here's an interesting fact. I don't know why I'm throwing this out there, but did you know that silicon-32 and radium-226, they actually shown that they changed from seasonal variation? Isn't that crazy? Their observation of decay rates actually changes when it's cold and warm.
So I think that it's easy to say that, though, because I can also say that certain elements, when you apply whatever, I believe it was electromagnetic fields, that they change their decay rates. But when you actually go in and look at the paper, the decay rate changes by less than 1%.
Yeah, it's almost
which is a hefty amount. And actually the guys at Rate for AIG and Creation Ministries and things like that, they were the ones that were like, yeah, look, we're going to have to assume some exotic solutions. We're going to have to look to miracles and things like that, or assume that we're going to find more about this in the future, because as of now, we got nothing, which is why I'm asking for the dates of this Ukrainian paper that you're talking about, because, and again, feel free to send it in the chat, that way, yeah, that way. Because I want to see the dates on this and see when it was published, because I would imagine that the geologists like Andrew Snelling, right, like he's like the go-to creationist geologist, even though in his conventional work he seems to accept billions of years for at least for his mainstream stuff, which he might have to for the job. But I would say, in addition to that, right, well, backing up, they would know about these papers and would probably have expounded quite a bit on them if they'd been released before the Rate project was disbanded. But moreover, I said this to Bill Morgan in our last discussion, companies all over the world use radiometric dating in order to find natural gases and oils. And they do, this is like a multi-billion-dollar industry that relies on this working. I'm sorry, go ahead.
I'm thinking I'm thinking that you're saying that I don't believe that the rates are correct. No, they can be 100% correct. That's the fascinating thing. The rates can actually be 100% true, but when they're created, they're already created with different parent to daughter isotopes already begun.
Right,
So,
but that's why I brought up isochron dating, which makes no assumption about the amount of parent material. So isochron dating it's a relatively recent method, I believe, in the past decade, and it makes exactly zero assumptions about the amount of parent material present at formation. And it still corroborates the dates that we get from traditional radiometric dating. You see what I mean? So I'm struggling to understand how you can make that claim. It sounds to me like, at least if I'm getting this correct in my head, that you're suggesting that, so if there are no radioactive elements prior to Noah's flood, which I don't know what a world, I'm not a physicist, I don't know what a world would look like if it had no unstable isotopes. I imagine that there would be quite a few natural processes impacted by that, but I don't know that for certain because, again, not a physicist. So you're saying that we only get radiometric decay beginning after Noah's flood, and then the elements that we have are only giving the illusion that they are actually very old, because when they were instituted or whatever in Noah's flood, there was actually quite a bit of daughter material already available. Am I correct in that?
correct. Yes.
Okay. So then my question with that would have to be, it's an awfully large coincidence then when we compare sort of our radiometric dating methods to plate tectonics. So the way that the plates move on top of the mantle, right, via convection, it's completely independent to anything regarding physics or radiometric decay, radioactive decay, right? It's literally just big slabs of continent on top of magma moving at a relatively steady rate. It has like a range, but it's generally like an inch or 0.8 inches, I think, to 1.2 inches per year that the continents are moving apart or together, depending on which one we're looking at. And if you look at the mid-Atlantic ridge, right, which is, of course, in the middle of the Atlantic, there's a big seaway there that's, or big whatever volcanic way there that's pumping out new continental shelves all the time through burbling magma, the ring of fire and all that. We all learned it in middle school earth science. But you can make a prediction, right? The rock that's furthest away from this burbling magma crack in the crust is probably going to be older than the magma that's sort of at the center of the crust, because it's coming out, at a relatively steady pace, at least from what we can tell since we've been measuring it. And so it's awfully strange. And of course, we get these rates by having like satellites taking pictures and things like that. It's awfully strange that when we take samples from, very close to this crack in the crust, and again, at the edges of this mid-Atlantic ridge, that they corroborate our radiometric dating. Like when we date the rock, right, via radiometric dating, and when we scale the rock at the edge all the way back using a similar rate of seafloor spreading, those things match. They're entirely independent from one another, and they match. So what you're essentially saying is that God, for whatever reason, decided to have the movement of the continental plates, the continental drift of the planet, match up in stepwise fashion with the radiometric decay rates and measurements that we pick up, just both independently equaling a very ancient Earth, or at least appearing to equal a very ancient Earth, despite having nothing to do with one another. I mean, you realize what an awfully large coincidence that seems like, right?
Well, think about it like this: when the great floods, the fountains of the deep broke forth in the mid-Atlantic ridge, that's where the majority of the force took place and started pushing the continents apart, or the Pangea continent apart, into what it is today. So what happened is, if this is where that event is occurring, and this electrical surge called the Z-pinch is flowing through granite rock, and the crystals that are in the rock are creating this force, and it's creating these new elements. What's going to happen then is there is going to be variances dependent on basically how far these things spread apart from each other. And like I was saying, that this study showed that when ththese elements were created, they're created instantly, and then all of a sudden there's dissipation. Right, they start decaying from their 100% into their weaker elements, into their parent to daughter. So if this is occurring and it's being spread out, by the way, God's not doing this. It's just, these are just elemental forces taking place on Earth, and so we can track those. So if it just because they are dating as though they're old, that's our interpretation of them dating to be old, because we're assuming that all of a sudden, well, it takes millions of years for this process to occur, but this experiment shows them occurring immediately. So that's the cool part about it.
So again, I do really want to see this paper, and I want to see the other one too. I don't think you sent it to me.
I have so many links. I'm not even sure which one it is, because as you can see here.
Just send them. I would send me all of them, and I'm more than happy to dig through them.
Okay. I just want to jump in real quick, guys. guys. Sorry. It's been a phenomenal discussion. Everybody's loving it. I'm loving it. We could probably listen to this all night. I do want to respect the debaters' times. Therefore, if you think this is fair, what we can do is kind of end it with, we can start with you, Erica, because you started, I think, in the intro. Give a couple minute summary. If you want to respond to something that Matt just said regarding the radiometric dating, you can. Just use the 2 minutes or so, however you please. We can do the same with Matt, and then we can call it a night if you think that's fair.
Absolutely. That's totally cool with me.
Beautiful.
Yes. So is that cool with you, Matt?
Oh yeah, fine. Go ahead.
Yes. So essentially, I'm going to take my time and I'm going to respond to what you just said. So again, you're using the Z-pinch method, which I'm going to, just for the sake of argument, assume that is 100% true. I don't think it is. I really want to see the paper, but I'm going to assume that it's true. Even if that is the case, I'm going to reiterate my past point. The rate at which, the seafloor is spreading is entirely independent of radiometric dating. That means it has nothing to do with physics. It's just basic, I guess it has to do with physics in that it's like Newtonian physics. It's just like movement, right? It's continents sitting on top of magma, and then the convection currents are what's moving the plates either to one another or apart from one another at roughly an inch per year. This is entirely separate from this Z-pinch that you're suggesting. So there is no reason to suggest that they would be stepwise, and that's why I said we're assuming that God would essentially have to intervene here and say, okay, boom, they're stepwise. Because otherwise you've got this weird situation, like a 1 in a trillion, where the Z-pinch occurred in such a way that it precisely matches up with not just the rate at which the seafloor is spreading apart, but also all of our ice core data, all of the dendrochronology, the coral clocks that we use, none of which have anything to do with radioactive decay, right? We measure coral clocks by growth rate and the size of the reef, the height of the reef. I think the Inuit atoll is usually used. Same thing with ice cores. It's regular snow rates, or dendrochronology, which is rings. I think creationists usually like to say sometimes you get double rings, but that's actually not the case. You're more likely to get sort of the opposite of what you would need, right? At least according to Wood-Marap, the creationist who has trouble with the bristlecone pines. But again, I feel like you're invoking kind of some funky physics here. That's again, why I want to see the paper for myself, and see when it was from. Because I would find it very interesting if it was from the past. Genuinely very interesting if it was from the past 5 years, because obviously I wouldn't have seen it, and the guys at Rate wouldn't have seen it. But if it's even a decade old or 15 years old, you sure would think that the guys at Answers in Genesis or Creation.com would have kind of sunk their teeth into it. And I keep decent tabs on those guys, and I haven't seen it. So to summarize, you have these multiple, 4 to 5 independent lines that have, for some reason, all converged on saying the Earth is very ancient. I think that's going to need a miracle invocation for that particular thing. And then 2, you have the guys whose sole mission is to evangelize young Earth creationism, not talking about it. So those are my 2 kind of, but please show me the paper moments. And that's all I have to say.
All right. I guess I'm up. All right. Well, yeah, I mean, we can always jump into that. I think that what is fascinating is that when the people go around the world and they look at these megalithic sites and they start testing them, they find all these discrepancies. That's why people invoke ancient aliens nowadays, because they're finding things that are before humanity even existed. So it's just what's going to happen sometimes, is because they're building these places with elements that were formed in the flood. So I think that's why there's discrepancies. And but I mean, coral clocks, their estimates are all right, but then when they find that the temperature in the ocean fluctuates, and the warmer it is, the faster they grow. I mean, it's okay, but it's not a really strong case in my mind. I think that I kind of showed that there's a line in ancestry that can be drawn, and I was shown a little bit. So I'll have a video out on that one coming soon, just to definitively kind of set it in stone. We're going to use more on this coming up. And that'll be good. And the reason you don't find these things in Answers in Genesis are things because they do not like Walt Brown. They don't like his theory, so they don't talk about it. That's why you've never heard of it. I recommend watching a video called by Brian Nichols, and he has it. It's called Where Did Earth's Radiometric Dating Come From? And I'll type it in right here for you. See how bad I am at debating. I'm just randomly conversating with you. And other than that, we covered a lot. So I don't think that a new anatomical feature arose in some lizard because it decided to become vegetarian, because we can find these cecal valves in other lizard species all around the earth that are geographically isolated. They're in different lizards besides these lizards, and they look exactly the same. They're just a muscle. I mean, epigenetics, I really wanted to get into like showing that all these beneficial mutations that were on in the past, when they talk about, well, there was a mutation where we found the lactose and it can be tracked to one woman, and then they found that wasn't true. It's just a methylation that happened inside that showed, oh, there's no beneficial mutation at all. And then the bone density was just another methylation event that occurred in somebody that worked out more. All these immunity from HIV, the high altitudes of the Sherpa people, all this information is already set into the genome, and it's expressed differently by the genes that are already inside of us. And evolution needs hundreds of millions of examples of beneficial mutations. And we have these eukaryotic organisms and bacteria that can live hundreds of thousands in generations, and we can irradiate them. We can give them, we can change their diet. We can change their environment, and they cannot do even the most minute steps besides change their fitness. And I know evolution believes that that can happen throughout millions of years, but we would love even just the slightest example of something going on like that. And when everything is just being epigenetically switched on and off and genes being lost, I see everything as the exact opposite of what is required for the theory to be true. So I'm more interested in answering questions. So I like answering the phylogeny challenge coming up, that where to draw the line, and that's my next focus. So stay tuned, everybody.
Okay, well, thanks so much, Matt. Thanks so much, Erica. Once again, thanks for giving us your time. As I said earlier, and I'll say it again, it was a great discussion. Time flew by, and the audience had a good time. So, of course, that's the most important part. So, once again, thanks so much for your closings, and we're gonna shut it down. So, Praise, it's up to you, brother.